So welcome back. So now we've just had a nice whistle-stop tour of all the things from DNA to RNA to protein and all the processes. So now we're going to move on to something that I find really interesting, which are mutations. So what we've covered in the previous lessons, how we go from DNA to RNA to proteins, how a protein folds, the consequences if a protein misfolds, and what can denature a protein and make it inactive. Obviously, if it's an enzyme and make it active, we've got problems. So now we're going to cover mutations. So like in the video, I hope you watched the one that I included as a bonus video, and that teaches us, and that just goes through some very quick points of how DNA mutations can occur. Now these can occur as a result of errors caused by external influences. However, some either occurred during development and weren't or couldn't be fixed, or they were genetically heritable. And it's these ones that we're going to be focusing on. So what are mutations and how do they affect protein structure? So you can have mutations at the DNA level, so obviously information goes from DNA to RNA to protein. So you can have mutations at the DNA level. Now this can be caused by genes or replication error. Now believe it or not, we actually do have checkers, and checkers actually go through our DNA sequence to check and make sure there are no errors. And unless they're checked, so not checked and not fixed, this can lead to problems. And unfortunately we see this kind of mutation in things like the BRCA1 gene, because BRCA1 and 2 are checkers. So they go through and check, and unfortunately when the proteins themselves, these guys are proteins, when these themselves are kind of not folded correctly, they have a problem checking DNA, which can obviously cause all kinds of problems. So we have mutations here, and it's often either a result of genes or replication errors. We also have mutations at the RNA level, so this would be a copying error. So RNA polymerase hasn't checked properly, and so instead of translating this, instead of transcribing this correctly, or a transcribing error. Okay, and in turn this obviously heavily affects here, and this is affected. Okay, so we can have errors at several levels. Now I'm not going to really be looking at errors at the mRNA copying level. I'm really going to be looking at your genetic errors, as it were. So we're going to be looking at genes. Okay. Okay, so what kind of mutations are there? So the ones I'm mainly going to be focusing on are indels. And they stand for insertions or deletions. Now below here, what am I doing? Now below here, I've got some key terms. We've got codons, indels, frameshift mutations, SNP. We've also got missense, nonsense, synonymous, non-synonymous, and codon redundancies. Now these are all key terms that you're probably going to need to know at some point, and a lot of them overlap. So the ones that we're going to be looking at, obviously a codon is something we've covered before, 3 base pairs that are read to make an amino acid. Indels are insertions or deletions. Now this is when we insert or delete a base pair. So we will do that one as an example. So I've already translated this here. So not translated, sorry, I've already put the base pairs along here. So if we translate that into codons, so our first thing is codons. And you've obviously got your UTR, 5', 3', UTR here. So we will translate this. So put that as an M, an I, an A, TAT is a T, A, T is a tyrosine, which is a Y. Then you've got arginine, histidine. So you've got arginine, histidine, and then that's a stop codon there. Okay, so TAA is a stop codon. So we've translated that into our amino acids. Now I'm going to give you an example of a SNP, or a point mutation. Now a SNP stands for single nucleotide polymorphism, which is simply that something here changes by 1 base pair. So what I'm going to do is I'm going to insert an extra one, and we will insert it, we'll insert that here. So we're going to make that C. Now this goes on to our next key term, which is a frameshift mutation, because this completely changes how we read the DNA. So frameshift mutation, a mutation that affects how the DNA sequence is read. So far up until this point, what's going to stay the same? Methionine is going to stay the same, isoleucine is going to stay the same, alanine is going to stay the same, tyrosine is going to stay the same, and now this is where our error happens. So we can say goodbye to arginine, histidine, and perhaps our stop codon. So you're going to have to forgive me because I'm going to have to kind of translate this as we go. So that's CCG, so we have a C, we have a C, and we have a G. So that makes that a proline now. We have then a GCA, so we have a G, a C, and an A. That makes that alanine. So then we have a CTA. We have a C, T and A, that's a leucine. Now our stop codon is missing. Now this leads the UTR to be transcribed, because this is what happens without the stop codon. So let's get rid of this because it's now no longer going to be untranslated. We're going to translate it, and let's say this is a load of nonsense. So we're essentially just going to make this a load of, we'll make it T's. So ATT, ATT, that'd be another isoleucine. And you see there's no stop codon. Now this is what we call a frame shift mutation, because it's caused the DNA, how it reads, to completely shift. We have completely changed how the DNA is read. As a result, we've altered the amino acids. Not only have we altered them, we've changed obviously their characteristics. So obviously proline is not the same as the other amino acid we've just changed, or potentially it is, in terms of characteristics, but what we've done is we've frame shifted. Okay, now because there is no stop codon, so if there is no stop codon, it will keep transcribing until it does hit one. Okay, so this results in really long, mostly unfunctional proteins. So, non-functional, non-functional proteins. Okay, so it will keep going. Okay, so we will just go back. So we'll change all this now. We're going to get rid of this. So we've already just demonstrated a frame shift mutation. Okay, so our next keyword, so we've done a SNP. So that, by the way, one insertion is obviously, it causes problems. So what happens if we now delete one? So let's remove, who shall be our victim? We're going to remove this C. So this is a deletion. And once again, everything stays the same until it hits this. And instead now, what am I doing? It'll read this, and it will keep going on, because obviously there is no stop code on there again. So GGC would be, GC is glycine, ACT is threonine, AAT is asparagine, and it will keep going again until it hits the stop codon, which will cause a really long non-functional protein. So we'll just get rid of that, and we'll put our sequence back to the way it was. Okie dokie. So our next one is a missense mutation. So a mutation that causes one amino acid to be substituted for another. Now this can be harmful or non-harmful. And we'll see synonymous and non-synonymous. So essentially synonymous is harmless and non-synonymous is harmful. So I did one I prepared earlier somewhere along here, and here it is. Okay, so I actually did one earlier, so it was originally a Y, so we're going to call this one, so if I get that was originally Y, so let's remember that. This one is a Y. Okay, so we're going to switch an amino acid, a base pair, sorry, so we're going to get rid of that T, and we've put an A there instead. So once again, everything stays the same. We've got a methionine, an isoleucine, an alanine, and then we hit here, and it's no longer a Y. It's TAA, and TAA is a stop codon. So now we have a problem. We have a stop codon. So it does not matter what's here, because it's not going to get read. When it hits the stop codon, nothing gets read after it. So I'll put that here. So premature stop equals no further translation, which equals a truncated, lightly non-functional protein. Okay, so I just thought that would be a really great example, and that would make that non-synonymous, it's harmful. So this would make it harmful, so non-synonymous. That's a very harmful mutation because that is definitely going to result in a truncated protein. Okay, so the next one is a nonsense mutation. So we've actually just covered that one, so we did that one a little bit prematurely. So a nonsense mutation causes a premature stop codon to be inserted and causes a truncated protein unlikely to be able to function correctly. So we've just covered missense and nonsense. So a missense mutation, we'll do a little bit of a different one. So let's get rid of that. Let's get rid of all of this and we'll start again. So we'll leave methionine the same. So AT&T, that stood for isoleucine. Now let's change that. Let's make it stand for leucine. So we'll pretend that that's a CTT. So we'll change this one to a C without actually changing anything. Okay. So that actually now makes it an isoleucine. So we can scrap leucine and it's an isoleucine. Now obviously everything after stays the same. You still get your stop code on there. That's absolutely fine. Now isoleucine being swapped for leucine is not a big deal. They are both polar. They're both non-polar, sorry. So they both have exactly the same characteristics. So swapping leucine for isoleucine often won't make a huge difference. Now that depends where it is. Obviously, if this amino acid is somewhere real important, so if it's an enzyme like a binding site or something, that could potentially cause enough of a problem that the protein doesn't function properly. That could genuinely be what happens. But if it's in a structural part of the protein, then it's unlikely to be causing a huge error. It's unlikely to cause a problem. And this takes me to our key points. So it matters where the mutation happens. If it affects the start codon, no transcription will happen. So if we change methionine, which is ATG, to ATA, and we called it isoleucine, so let's call that one A, and it's isoleucine, well, it's just going to keep going. Obviously, it still binds when it comes to transcription. So the RNA polymerase still binds to the upstream promoter, but we have a problem and transcription won't initially begin. Okay? So it affects the start codon, no transcription will happen. If it affects the stop codon, transcription will keep happening until it finds a stop. Changes in amino acids that form part of the structure, you may have a little bit of flexibility. So I've already mentioned, if the change from leucine to isoleucine was in the structural part of the protein, you might be able to get away with it. If it happens in part of the binding site, so changes in amino acids that are part of the binding site are likely to cause problems. And it matters how it affected the amino acid. If the mutation doesn't change the amino acid, no problem. And this brings me on to codon redundancies. So you will see that here we have two codons that code for phenylalanine. You have got a load of codons that code for leucine and a fair few that code for isoleucine. Only one that codes for methionine and things like this. And this is what we call codon redundancies because we have loads of potentially different codons coding for the same amino acid. So if we go back to GCC, let's have a look at GCC. So GCC, that's alanine. So if I was to change that to GGA, so let's change that to GGA, it'll still be alanine. It doesn't matter. It does not matter that the mutation changed. It's still alanine. Okay? So that's what I mean by codon redundancies. So this brings me to the point of if the mutation doesn't change the amino acid, then no problem. You won't notice. If the mutation does cause a change in amino acid, but the amino acid is very similar to the original one, it's unlikely to make a huge difference, which obviously brings us to our point up here. If the mutation causes a change in amino acid that has a very different characteristic, so non-polar to polar, that's probably going to cause a problem. Again, you may have some flexibility in the structure, it all just depends. But essentially, it all comes down to whether it makes any kind of difference or not, and where it is that amino acid is meant to be. If it's a very, very important amino acid, you know, it's in the binding site for something, then you probably have very little flexibility with what mutation you can kind of get away with. So that just basically covers mutations. Once again, I'm sorry it's a whistle-stop tour, but I will be posting in the extra resources loads of other videos and things like this, and there's always YouTube, people go into this using animations and things like this, and that might help you understand everything in a little bit more detail. If you have any questions, do feel free to post this, obviously, on the forum. So I will see you in the practical, guys.